Good, that meme is dead anyway. Hello, I am Fuiwogfu. But you can call me Fuiwogfu. I am a bacteria. Inside me is something called CRISPR-Cas9, which helps me become stronger and eventually strong enough to wipe out all dead memes. CRISPR-Cas9 captures parts of attacking memes and uses them to recognize and destroy said memes when they attack. The first description of CRISPR was from Osaka University researcher Yoshizumi Ishino in 1987, who accidentally cloned part of a CRISPR together with the IAP gene. A major addition to the understanding of CRISPR came with Janssen's observation that the pbhqg repeats cluster was accompanied by a set of homologous genes that make up CRISPR-associated systems, or Cas genes. In 2005, 3 independent research groups showed that some CRISPR spaces are derived from phage DNA and extrachromosomal DNA such as plasmids. The spaces are fragments of DNA gathered from viruses that previously tried to attack the cell. The source of the spaces was a sign that the CRISPR-Cas system could have a role in adaptive immunity in bacteria. Researchers studied a simpler CRISPR system from Streptococcus pyogenes that relies on the protein Cas9. The Cas9 endonuclease is a 4-component system that includes 2 small RNA molecules named CRISPR-RNA and trans-activating CRISPR-RNA. Jennifer Doudna and Emmanuel Charpentier engineered the Cas9 endonuclease into a more manageable 2-component system by fusing the 2 RNA molecules into a single guide RNA that, when combined with Cas9, could find and cut the DNA target specified by the guide RNA. Baron Gao and Horvath showed that Cas9 from the S. thermophilus CRISPR system can also be reprogrammed to target a site of their choosing by changing the sequence of its crRNA. Feng Zhang's and George Church's groups simultaneously described genome editing in human cell cultures using CRISPR-Cas9 for the first time. It has since been used in a wide range of organisms, including baker's yeast, zebrafish, fruit flies, nematodes, plants, mice, and monkeys and human embryos. The CRISPR-Cas9 system has shown to make effective gene edits in human tripronuclear zygotes. The system made a successful cleavage of mutant beta hemoglobin in 28 out of 54 embryos. 4 out of the 28 embryos were successfully recombined using a donor template given by the scientists. The scientists showed that during DNA recombination of the cleaved strand, the homologous endogenous sequence HBD competes with the exogenous donor template. DNA repair in human embryos is much more complicated and particular than in derived stem cells. It is not perfect, however. In one study, researchers sequenced the entire genome of the CRISPR-edited mice to search for mutations. In addition to the intended genetic edit, they found more than 100 additional deletions and insertions along with more than 1,500 single nucleotide mutations. Many believe it is still too dangerous to use with humans. My name is Fuiwogfu, and this was CRISPR-Cas9.
I will build a great wall on our southern border, and I will have Mexico pay for that wall.